Okay. Looks good.
Okay, great. Yeah, I'd really like to thank Active Motif for putting on this Chromatin conference and also for being a really important local supporter of the San Diego Chromatin Club, in particular Seth Rubin. We've been doing this as a happy hour and then converted it into a very robust virtual seminar series, and they've been great in supporting us throughout that effort. So thank you. So yeah, just to tell you a little bit about the work that we do in my lab, like many of you, we are interested in the regulation of eukaryotic transcription. And as we've been discussing, this involves multiple different layers of epigenetic regulation, including, for example, the regulation of histone modifications, as well as things like 3D local chromatin structure. But our lab is primarily focused on the role of nucleosome occupancy and the contribution of chromatin remodeling complexes to generate open chromatin at cis-regulatory elements for transcription factors to bind and the binding of other epigenetic machinery. And so in particular, our lab is focused on the switch SNF or BAF chromatin remodeling complex, here illustrated based on several recent structural studies. So what we know about this complex is that it's composed of a core ATPase, either BERG or BERM, and several associated subunits. And an important feature of this complex is that many of these subunits are members of gene families that can combinatorially assemble to give rise to many different variants of the complex with distinct biological roles. And this was really elegantly shown in a number of mouse genetic studies in which it was shown that the subunits of the BAF complex are required at the very earliest stages of development, for example, for implantation of the blastocyst and the maintenance of embryonic stem cell pluripotency, as well as in the development of several different major organ systems, including the muscle, the heart, and the brain. And so perhaps not surprisingly, it was also shown that this complex is highly mutated in human cancer. So as a postdoc, my colleagues and I estimated that subunits of this complex are mutated in upwards of 20% of all human cancers. And this was in fact based on fairly limited sequencing data. But as more sequencing data has come to light, this figure has really stood the test of time. And we now know that ARID1A, for example, is the third most frequently mutated tumor suppressor, and PBRM1 is among the top 20. And so this has just spurred an incredible interest in understanding how this complex is regulating both normal development as well as disease and how mechanistically this complex is working. So when I started my lab, there were several sort of outstanding questions, including, does the complex regulate chromatin accessibility in vivo? And if so, what sites are affected? How is the complex targeted to sites of action? How does the complex control gene expression? And what is the relationship with transcription factors? How does combinatorial assembly contribute to biological function? And how do mutations in subunits of the complex give rise to disease? And so it's been a really exciting time in the field based on several technological advances that have allowed us to address these questions for the first time. So for example, the development of ATAC-seq by William Greenleaf and Howard Chang now gives us a really robust and cheap way to assay for switch SNF complex function at genome-wide scale. As a field, we've overcome difficulties in performing ChIP-seq for these large complexes that are prone to fragmentation, low antibody affinity, and indirect binding to DNA, really allowing us to detect the targeting of the complex and the function of the complex through these two different modalities. And I'll emphasize that the sequencing of human disease, both in cancer and in intellectual disability disorders, have revealed mutations in subunits that were not extensively studied in developmental models. And so it's really allowed the field to focus on specific subunits, specific missense mutations, and haploinsufficiency as drivers of disease. And finally, there has been a huge development of chemical inhibitors and degraders that have allowed us to acutely perturb the complex function to ask what are the direct effects of blocking complex function. And so getting to this first question, one of the first experiments we did was to assess chromatin accessibility in cells that were either wild type or lacking components of the BAF complex ARID1A and ARID1B. And we found indeed that using this ATAC-seq method, we were able to identify over 12,000 sites that lost chromatin accessibility an additional 5,000 sites that gain chromatin accessibility, really suggesting that this complex is a major regulator of open chromatin in general. However, what was perhaps more surprising is that this was really prominent at only a select number of sites. And this is readily observed when we cluster sites on the basis of accessibility in the wild type cell. So what we observed were there's two prominent clusters that were particularly sensitive to BAF complex deletion. And these clusters were highly enriched for BAF complex binding as defined by the BRG1 ChIP-seq here, and also had a major enrichment of other features of enhancer elements. And so collectively, my lab and other labs have converged on this model of switch-SNF complex function shown here, where we believe that the complex is highly enriched in enhancer elements, where it is really critical for maintaining essentially all aspects of enhancer architecture. And so that in the absence of the complex, we not only see a loss of chromatin accessibility, but this is followed by a loss of transcription factor binding, a loss of active histone modifications, eRNA synthesis, and ultimately loss of gene expression from the nearby promoter. We think that this activity of the complex really underscores its role in development and cellular differentiation, as this is what gives rise to cell type specific gene expression in these contexts. And so given that this complex function was actually quite selective, we were curious as to how this complex was being recruited to chromatin. And because the complex doesn't have any sequence specific DNA binding activity, we were quite excited by the idea that various transcription factors, including lineage determining transcription factors and signal responsive transcription factors, might play a role in recruiting the complex to chromatin. But we were also aware that several subunits of the complex contain protein domains that interface directly with chromatin, including bromodomains, chromodomains, DNA binding domains, and Ph.D. domains. And so we were wondering what the contribution of these domains might be to complex targeting and the maintenance of complex binding. So we've specifically focused on a number of subunits that contain this bromodomain protein domain. So this is the bromodomain family tree, and I'll just point out several BAF complex subunits on this tree. There's the SMARCA4 and SMARCA2 ATPases, which are highly related to the bromodomains in the polybromo1 subunit. And then over here, there are two other bromodomain subunits, the BRDi and highly related BRD7 bromodomain subunit. And again, these are quite distinct from the more well-known BET family of proteins encompassing BRD2, 3, and 4. And so we initially focused on this BRD9 subunit because there were already several inhibitors that have been developed that could block the binding of the BRD9 bromodomain to acetylated residues. And so we thought that this might be a really facile way to assess the role of the BRD9 bromodomain in complex targeting. But it was in our first sort of set of standard experiments that we got our first surprise, and that is through an IP mass spec of BRD9 interacting proteins, in which we recovered many BAF complex subunits as expected, but importantly, not all. And we also revealed association with two proteins, Glitzker-1 and Glitzker-1-like, that we had not previously focused on. And so to see if this was an issue of detection, we performed IP Western blot, and you can see that an IP of the BRG ATPase reveals association with all of these different subunits. An IP with BRD9 reveals association with several BAF complex members, but not ARID1A or BAF47. BAF47, a reciprocal IP of BAF47, brings down ARID1A, but not BRD9 or these two new proteins, Glitzker-1 and Glitzker-1-like. And so this really started to look like it might be a unique entity. And indeed, when we perform glycerol gradient sedimentation analysis, which we typically use to separate complexes on the basis of size, we indeed found that BRD9 and Glitzker-1 were migrating at these smaller molecular weight fractions, quite apart from the known BAF and PBAF complexes, and importantly, lacked association with ARID1A, BAF47, and BAF57. And so collectively, work from my lab and several other labs, we've defined a new variant of the switch-SNF complex, now referred to as the non-canonical or NC-BAF complex, also known as the GBAF complex. And this was quite exciting, got the field aflutter, in that there hasn't been a new complex identified for at least 20 years. And really focused our attention on, again, this idea of combinatorial assembly and the idea that the incorporation of unique subunits, including BRD9, might give rise to different targeting and functional properties of these complexes, which would distinguish them in terms of transcriptional regulation. And so that's really been the framework that we've been operating under to understand the function of these different variants. And one way we began to do this was to approach it using ChIP-seq to assess the binding of these different complex members. And so we performed ChIP-seq, for example, for ARID1A to localize the canonical complex, and BRD9 to localize the non-canonical complex, as well as the BRG1 ATPase. And we found that while ARID1A and BRD9 both overlap quite well with BRG binding, they actually have pretty unique binding patterns, and suggesting that there were very specific binding patterns of these two different complexes. And indeed, when we look a little deeper, we find that BRD9 is more highly enriched at promoters, whereas ARID1A seems to be more enriched at these distal sites. Taking a deeper dive into histone modifications, we find that ARID1A sites are enriched for H3K4 monomethylation, often found in enhancers. And indeed, ARID1A is more highly enriched at essentially all classes of enhancers, including poised, active, and super enhancers. Whereas BRD9 appears to be more strongly enriched at sites decorated with H3K4 trimethylation found at promoters. In addition, we found that BRD9 uniquely is enriched for binding of both cohesin and CTCF. And this appears to be a unique property of BRD9 because ARID1A was fairly deficient in the binding of these proteins. So pairing up with Jesse Dixon, we found that BRD9 was actually highly bound at these so-called TAD boundary elements. And again, this was a specific property of BRD9 because ARID1A effectively had no binding at these elements. And so this really started to put together a framework for us to understand how this biochemical diversity among the different variants could give rise to these different recruitment and targeting strategies that could give rise to different functional properties of these complex variants. So as I mentioned, we think that the canonical form of the complex is highly enriched in enhancers, where it's important for enhancer activity. Here we find that the non-canonical complex is enriched at promoters and CTCF bound sites. And there was other information to suggest that PBAF complexes were bound at promoters as well. And so this really started to suggest that these complexes might be affecting transcription in different functional ways. And so to assess this, we turn to, again, the wealth of inhibitors and degraders that have been developed against these complexes, some of which allow the specific targeting of these variant-specific forms. And so I'll just draw your attention to the putative ARID1A inhibitor developed by the Dijkhuizen lab, as well as several BRD9 inhibitors that again bind the BRD9 bromodomain and effectively inhibit it from binding acetylated residues. And also this degrader dBRD9 molecule, which acts as a heterobifunctional molecule by binding the BRD9 bromodomain and targeting it for the proteasome. In addition, there is this BRG-BRM degrader, which is effectively wipes out all forms of the complex. And so using these chemical inhibitors, we can look at ways to acutely disrupt the function of these different variants. And so our approach is really just to use ChIP-seq to basically identify the binding patterns of these different variant functions, and then to use either genetic deletion or chemical perturbation to ask what the effect of disrupting these complex forms is on both transcription and ultimately cellular biology. And so I suppose you could potentially study BAF complex function in essentially any cell type, but we're really attracted to the idea of understanding BAF complex function in various immune cell types, including macrophages and T cells. And one of the reasons why we study macrophages is because they're really robust in vitro systems to understand the role of the BAF complex in cellular differentiation of macrophages and in activation of genes in response to inflammatory signals. And so, for example, following stimulation with lipopolysaccharide or lipid A, macrophages are activated through the TLR4 receptor, which leads to the activation of MyD88 and TRIF, which then leads to the activation of these transcription factors, NF-kappa B and IRF, which leads to the very rapid induction of cytokines and chemokines, as well as the expression of interferon, type 1 interferons. And so we felt like this might be a great way, as it has been used in the past, to understand the role of the BAF complex in inducible transcription. And so again, we made use of these BRD9 inhibitors to specifically assess the role of BRD9. And we wanted to not only understand the contribution of BRD9, but assess the effect of these different inhibitors on BRD9 complex function. And so we chose iBRD9, this BI9564 compound, and dBRD9 to compare. And the way we set this up was essentially to treat macrophages for 24 hours with these BRD9 inhibitors, and then stimulate for 1 or 4 hours with lipid A, and then to assess the effect on transcription through RNA-seq, and then perform ChIP-seq for BRD9 and other associated factors. In addition to BRD9, there was quite a bit of evidence that was mounting to suggest that BRD9 and the non-canonical complex might be cooperating with BET proteins. So I'll remind you, these BET proteins, for example, BRD2, 3, and 4, are important players in the regulation of transcriptional elongation, super enhancer activity, and phase separation. And so really have broad and pervasive roles in transcription. There's mounting evidence that there is a very strong biochemical interaction between BRD9 and both BRD4 and BRD2. And in addition, inhibition of BRD9 and inhibition of BET proteins leads to a very similar effect on gene transcription. For example, in data that we showed in mouse embryonic stem cell pluripotency, and data from the Dijkhuizen lab in androgen receptor dependent genes in prostate cancer. And so based on this potential cooperation between BRD9 and BET proteins, we included a BET inhibitor in this series just for comparison. And so here I'm showing the differentially expressed genes following lipid A activation in the presence of each of these different BRD9 inhibiting agents. And so what you can appreciate is that independent of what agent we use, there is a common core set of genes that are affected by all 4 agents, really suggesting that BRD9 has an important role in regulating lipid A-induced transcription. What you can also appreciate is that dBRD9 and BI9564 have a highly overlapping gene signature, which is comparatively less potent than that of iBRD9 or JQ1, which again affect many more genes and converge on a common set of genes that is not affected by the other 2 inhibitors. And so this started to look like these inhibitors might have different mechanisms of action, and we were curious as to what those might be. And so we performed ChIP-seq for both BRD9 and BRD4 in the presence of these different agents. And here I'm just summarizing the data. I'll start you over here looking at the treatment with BI9564. So we found that treatment of cells with BI9564 led to displacement of BRD9 at over 6,000 sites, really suggesting that this bromodomain inhibitor acts to displace BRD9 in the non-canonical complex from chromatin. And in contrast, had a limited effect on BRD4 binding. dBRD9 acts to completely degrade the BRD9 subunit, and in response, we effectively saw complete widespread loss of BRD9 binding to chromatin in these treatments. And again, a very limited effect on BRD4. In contrast to these agents, both iBRD9 and JQ1 led to the loss of both BRD9 and BRD4, and there was really no selective advantage for either of these proteins with either agent. And this really correlates with the effect of these agents on gene expression. So as I mentioned, iBRD9 and JQ1 have a really robust effect on gene expression. And we don't know, we can't really ascribe that to BRD9 because these agents are hitting both BRD4 and BRD9. In contrast, dBRD9 and BI9564 appear to map to the function of BRD9 quite well, and again, have a robust effect on inducible gene expression. And so going forward, we wanted to understand how BRD9 was regulating gene expression, and in particular, which genes. And so we turned to several public data sets in which various components of this pathway have been deleted to understand which gene sets BRD9 is affecting. And so I'll remind you that stimulation with LPS or lipid A gives rise to the activation of MyD88 and TRIF, which activates this series of transcription factors, which translocate to the nucleus to transcribe multiple genes, including interferon, type 1 interferons. Interferon beta can be translated and secreted and signal in an autocrine manner through the type 1 interferon receptor, which activates the so-called ISGF3 complex composed of STAT1, STAT2, and IRF9. This ISGF3 complex can also translate to the nucleus to bind IRF elements and ISRE elements to activate ISGs, or interferon stimulated genes. And so by knocking out components such as IRF3 and the type 1 interferon receptor, several investigators have shown that the majority of the secondary response to lipid A is driven through this type 1 interferon receptor. And indeed, when we look at the genes that are affected in response to BRD9 inhibitors, we find that a number of these genes are also affected in the absence of IRF3 and the interferon receptor knockout, really suggesting that BRD9 is potentially affecting this interferon receptor pathway. And consistent with this, many of the genes that we found to be BRD9 dependent are highly enriched in genes of the interferon gamma response and interferon alpha response, these so-called ISGs. And so to understand how BRD9 is regulating the expression of ISGs, we performed ChIP-seq for BRD9 in either untreated cells or lipid A stimulated cells. And we saw a dramatic increase in BRD9 binding in response to lipid A stimulation at several thousand sites. At these gain sites, we also saw an increase in BRD4 binding. And when we looked specifically at the co-gain sites occupied both by BRD9 and BRD4, we saw a strong enrichment of this IRF motif, suggesting that these epigenetic regulators may be recruited by the ISGF3 complex. And indeed, we see a robust binding of IRF9, STAT1, and STAT2 in response to lipid A at these BRD9 gain sites. What was interesting to us is that when we looked at the co-binding of all these factors, we found that there was a dramatic percentage of peaks that were found at promoter elements relative to the binding of either BRD9 or BRD4 alone. And so in the presence of lipid A and interferon beta, we really see a recruitment of BRD9 and BRD4 to these promoters. And in fact, when we look at promoters that are co-bound by both BRD9 and BRD4, over 70% of these actually have ISGF3 binding in response to lipid A or interferon beta. And so you can see this quite nicely at this ISG cassette here, where there are multiple ISGs that are activated. And you can really see the very highly regulated binding of ISGF3 at the promoters of these genes. And in response to stimulation, we also see recruitment of BRD9 to these promoters and recruitment of BRD4 to these promoters. And this is distinct from the binding of BRD4 that occurs in a non-regulated fashion, for example, at non-inducible genes. And so we wanted to understand, of course, how BRD9 might be regulating the transcription of these genes. And so we went back to perform a TAC-seq in the presence of the BRD9 inhibitors. But similar to our experience in multiple other cell systems, we found that there were very few changes in a TAC-seq upon inhibition with BRD9 inhibitors, really suggesting that BRD9 may have a distinct functional role in controlling the transcription of these genes. And so we turned to the regulation of this transcription factor binding, specifically ISGF3. And we performed ChIP-seq on IRF9 or STAT2 in unstimulated cells or in response to stimulation, plus or minus treatment with each of these inhibitors. And you can see that both IRF9 and STAT2 have a very robust induction of binding, but that this binding in response to lipid A is generally reduced in the presence of these inhibitors. And again, there is this distinct effect where BI9564 has a relatively limited effect on the binding of these factors, where DBRD9, IBRD9, and JQ1 really lead to almost the complete obliteration of binding of these factors. And this is seen here in these histograms of the average signal, again, the induction of this binding and overall reduction in response to inhibitor treatment. So linking this back to gene expression, we're curious to know how often the disruption of this binding led to changes in gene expression. And so we looked at our differentially expressed genes in each of the different inhibitor treatments and asked how often a gene experienced loss of binding of IRF9, STAT2, or STAT1. And just identified by these blue pies here, you can see that the vast majority of genes experience loss of at least one of these factors in response to BRD9 inhibitor treatment. And again, you can see that quite well on the genome browser. So I'll just focus your attention here to BRD9. Again, we see this stimulated recruitment of BRD9 in response to lipid A, which is lost in the presence of DBRD9. And in response to DBRD9, we also see a reduction in essentially all of these factors, IRF9, STAT1, and STAT2, at both these BRD9 dependent genes. And so this really validated and confirmed for us much of our hypotheses that this biochemical diversity could lead to different functional roles. So again, the canonical complex we think is enriched in enhancers where it is recruited by transcription factors and serves to maintain chromatin accessibility for transcription factor binding. The non-canonical complex, in contrast, we believe is recruited by either general transcription factors or signal responsive transcription factors, here ISGF3, bringing these complexes to chromatin. However, we do see an important role for the BRD9 bromodomain in that inhibition by BI9564 leads to displacement of the BRD9 protein. And thus we believe that this bromodomain is important for either recruiting or stabilizing the complex on chromatin. Importantly, we see again this cooperation with BRD proteins, BRD2 and BRD4, in that inhibition of BET proteins and inhibition of BRD9 lead to very similar effects on transcription. And this leads us to posit that the non-canonical complex might have important roles in facilitating various BET-dependent processes. And the fact that these two proteins were found recruited to promoters makes us wonder whether BRD9, for example, might be important for facilitating transcriptional elongation through BRD4, which is an important BET-dependent process. And that's something that we're currently interested in pursuing. So taking you back, we're essentially trying to fill in this sort of features table of the different variant forms and really highlighting how this biochemical diversity and the incorporation of unique subunits can give rise to different functional attributes of the complex, which may have different impacts on cellular biology. So as I mentioned, the genomic targeting, we've observed that this is distinct for both the BAF complex, non-canonical BAF complex, and PBAF complex. We found different mechanisms of recruitment, including both transcription factor dependent recruitment, as well as the contribution of bromodomains. As I mentioned, the role of the canonical complex is in traditional chromatin remodeling and recruitment of P300 to enhancers. In contrast, we see very little action of the NC-BAF complex in generating chromatin accessibility. And so we are now pursuing a possibility that the NC-BAF complex might be cooperating with BET proteins or indeed cooperating with CTCF and cohesin to regulate chromosome topology. And again, this leaves sort of an open question as to how these complexes are regulating transcription. And again, I think we know sort of even less about the PBAF complex in terms of its functional attributes and features. So finally, switching gears, I just want to give you sort of a quick glimpse at how we are approaching this in more complex in vivo systems. And our attention is really focused on the tumor immune microenvironment, which you may be aware is composed not only of tumor cells, but several immune cell types, including cytotoxic CD8 T cells, which are important for tumor cell killing, as well as several immunosuppressive cell types like T regulatory cells and tumor-associated macrophages. So the immune cell types within the tumor microenvironment are constantly being activated and polarized in response to factors secreted by the tumor. And so we posit that BAF complexes, just as they are responsive to inflammatory stimuli, are likely responsive to these environmental signals and have a role in polarizing immune cell types within this microenvironment. And so in that way, we hypothesize that the BAF complex is contributing to shaping the tumor immune microenvironment, and that modulating BAF complex function could have either advantageous or deleterious effects on the tumor immune microenvironment. And this is sort of an exciting concept for us, because it's certainly, if we could modulate BAF complex activity to create a more immunogenic or immunostimulatory immune microenvironment, we'd certainly like to know. And so I'll just tell you about one collaboration that we have ongoing at the Salk Institute with my neighbor, Ye Tseng. So Ye is a expert in T regulatory cells. These cells are important suppressors of T effector function. And so they are really important for inhibiting a broad range of autoimmune diseases. But as I mentioned, they're also a really critical barrier to effective tumor immunity. And so for that reason, there's a lot of interest in being able to down-modulate the activity of Tregs for cancer therapy. So Tregs are largely defined by the expression of this FOXP3 master regulator. And indeed, their suppressor activity is tightly correlated with the expression of FOXP3. And so in a genome-wide screen that Ye performed, he found many BAF complex subunits were important for regulating the levels of FOXP3, which really sparked a nice collaboration between our labs. And so I'll just draw your attention to some of the secondary screens that we performed. So here we are showing essentially Tregs that have been transduced with guide RNAs against subunits of the BAF, non-canonical BAF, and PBAF complexes, and then measuring the levels of FOXP3 expression by flow cytometry. And what you can see is that modulating subunits of the BAF complex has very little effect on FOXP3 expression. Whereas if we deplete subunits of the NC-BAF complex, including BRDi, we see a really robust downregulation of FOXP3. And when we deplete subunits of the PBAF complex, we see a slight upregulation of FOXP3. So in this study, we revealed this sort of important antagonism between NC-BAF and PBAF complexes in controlling the expression of this master regulator, in that NC-BAF complexes positively regulate the expression of FOXP3, whereas PBAF complexes negatively regulate this expression. And so really suggesting that we could use modulation of these complexes to toggle FOXP3 expression and thereby toggle Treg suppressor activity. And then coming to, again, our favorite BRD9 inhibitors, we found that incubating Tregs with dBRD9 results in a dose-dependent decrease in FOXP3 protein expression. And similarly, incubating with dBRD9 results in a loss of suppressor activity, as shown by this in vitro suppressor assay. And so again, this suggests that chemical modulation of BRD9 could be effective in reducing Treg suppressor activity, which might be important for igniting immune cells in the cancer microenvironment. And so to test this, we made use of an MC38 colorectal cancer cell line, which can be injected into the flanks of animals. And so we did this in animals that are either wild type or depleted for BRD9 in the Treg compartment. And you can see that injection of this cell line results in uncontrolled cancer growth in the context of wild type animals. However, when we deplete BRD9 and Tregs, we find that this reduces the suppressor activity, and this cancer growth is effectively controlled by the action of CD8 T cells. And here we find an increased infiltration of CD8 T cells, and these T cells are secreting more interferon gamma, more perforin, and more granzyme. So effectively, by depleting BRD9 in the Treg compartment, we've converted this cold microenvironment into one that is now immunogenic and immunostimulatory. So this is really kind of providing us a framework with which to approach many different cell types within the tumor microenvironment. And so what we've done is really develop mouse models in which we can, we've deleted specific BAF complex subunits in specific immune cell types and understand how they affect the tumor immune microenvironment. We've also perfected several functional genomics, such as techniques such as cut and run and cut and tag, which can be used to target BAF complexes and assess their functional activity on these very low numbers of cells coming out of the tumor. And finally, we're taking this unbiased approach by injecting switch-SNF complex inhibitors and asking what is the effect on the tumor immune microenvironment in the context of this inhibitor therapy. And this is obviously something that's quite important because as these inhibitors advance to preclinical and clinical trials for cancer therapy, we really have very little understanding of the effects on immune cell function. And so understanding the properties that these inhibitors have on immune cell function could be quite important for their synergy, for example, in cancer therapy, or their use in other immune dysfunction disorders. And so finally, I just want to thank my lab. I want to particularly thank Jovel and Gachalian, who was spearheaded the work on the discovery of the nBAF complex and also the collaboration looking at the role of BRD9 in T regulatory cells. And she was assisted by my lab manager, Josephine Ho. And then I also want to thank Nasia and Ahmed, who really did a lot of work on the macrophage system to identify the role of BRD9 in working with ISGF3. And again, my collaborators, Ye Zhang, as well as Jesse Dixon and Ron Evans, all here at the Salk Institute, and my sources of funding. So I'll just stop there. Take any questions.
Thank you. Thanks, Diana. All right, well, yeah, we already have a couple questions and I'm not surprised. It was a fantastic talk. So first we have one from Francesca saying, thanks for the interesting talk. Do you have any idea on how BRD9 affects IRF9 STAT1-2 recruitment upon lipid A stimulation without affecting DNA accessibility?
Yeah, that's a good question. And we don't really know what the action of BRD9 complexes is in controlling chromatin accessibility. So we haven't been able to see major changes by attack-seq. And it's possible that that is something we need to explore by MNA-seq or something with higher resolution. But I think there's also a fair amount of evidence to suggest that the non-canonical complex may be affecting chromatin accessibility in different ways, because it doesn't include some of the core subunits that are really required for chromatin remodeling. And so I think there's a lot to learn about how this complex might be acting and again, how it might be stabilizing transcription factors as we found. It's a good question.
All right, and then Bing Ren asks, it's very interesting to see BRD9 and nBAF complex co-localized with CTCF. Does nBAF regulate CTCF binding or chromatin looping at promoters?
Yeah, so we know, and this is really nice work by Dirk Schubler, that in general, the BAF complex through knockout of BRG1, the ATPase, does not regulate constitutive CTCF binding. But I think what's less well appreciated is whether BRD9 and the nBAF complex might have a role in inducible looping. And I think that's something that has been shown, but needs more attention. Yeah, we really need to look at it a little more closely. And this is a good system to do that in.
Cool. I had sort of a really simplistic question while other people are writing questions in there, but I really liked how you started out with chipping each of the different factors and seeing that, sure, in general, they all bind together as we'd expect, but the different sites where they were sort of moonlighting on their own. Have you looked at ProSeq data sets in any of these cells? Because I know the